Beyond using Google Maps and consulting your cell phone, have you ever wondered how we successfully navigate our environment in our day-to-day lives?
In this study, Doeller and Burgess chose to explore how humans are successfully able to navigate their environment by testing the memory of paths that they have traveled before. They were specifically looking for the presence of grid cells in humans using classic fMRI techniques combined with new technology.
It turns out that we may have an internal network of cells responsible for encoding our path information and our understanding of our location in space. These are known as grid cells and are encoded by speed and angular orientation. It is thought that these cells represent space as a grid of equilateral triangles and fire sequentially in relation to one's position in the environment. In addition, it's thought that your speed and angular orientation correlate to the strength of firing in this network. For example, when you cross the street, your grid cells may fire in a path similar to this one. Oh, my coworker has arrived!
Oh, are you talking about grid cells?
We are.
It turns out these cells were first discovered in rodents using electrode recordings of the medial anterorhinal cortex. These cells were seen to fire when the rodent moved in a direction aligned with a triangular grid. However, there's yet to be proof that these cells exist in humans, and if they do, where they're located in the brain. To exactly repeat this experiment in humans, it would mean attaching an electrode to a human brain in vivo and having them navigate in a defined environment.
For obvious reasons, we couldn't repeat the same procedure used in rats.
So how would we study this in humans?
I'm glad you asked.
Doeller and Burgess hypothesized that they could use virtual reality to mimic grid cell-like activation in a real environment, and subsequently use fMRI to detect the resulting bolt response. Because these cells do fire individually and not as a population, it would be hard to detect these cells when we consider the limitations of voxel size. However, Doeller and Burgess hypothesized that grid cell firing could be detected in fMRI based on three factors. 1. Angular orientation of the grid is relative to the environment across cells, such that each individual has a different defined grid orientation. 2. Conjunctive cells firing is modulated by running direction. 3. It was hypothesized that the speed of movement would correlate to firing frequency rate, and ultimately the intensity of neural activation in a voxel. The first two factors discussed will create systematic differences in neural populations, allowing for the detections of pathways that are aligned or misaligned to the main axis of the grid.
To test this hypothesis, human participants mimicked a foraging task using virtual reality. Their goal was to collect items using handheld computer key controls, and in a subsequent trial, replace these items to their original location. The goal was to measure the accuracy of spatial memory based on how closely participants were able to replace the item relative to its original location. The virtual reality landscape included geographical cues to inform both the participants' orientation and the location of the object within the arena. The 42 participants in the study were all male, aged 18 to 31, were all right-handed with normal vision, and had previous experience with video games.
A 3D scanner was used to collect the data, which was then pre-processed for spatial realignment, correction for differences in slice acquisition time, spatial normalization, and smoothing. An ROI approach was used to locate the anterorhinal cortexes of each participant. A set of GLMs was used to model the data. Half of the MRI data, the odd trials, was used to make a GLM for each participant. This was used to determine the overall orientation of the potential grid cell network. This was observed by the presence of fMRI adaptation modulated by running direction. The model included a predicted factor of 6-fold rotational symmetry in the grid cell network. This is the grid that was proposed in rats, and the study is looking to see if there is a similar grid in humans.
Consider the following. We know that in fMRI studies, when an action is performed repetitively, reduced activations of an involved neural population will be observed. In this case, when a participant repeated their path in the VR arena, decreases in activation of neural populations encoding for that running direction was observed. The information could be used to determine a possible overall orientation for each participant's grid network.
This model for orientation was applied to the second half of the data to determine differential activation for runs that were aligned versus misaligned to the primary axis of the grid. It was hypothesized that activation would be modulated by running speed, where faster running would result in higher activation as denoted by the blue line. I have the results.
Oh, let's take a look. I see here that fMRI adaptation was observed with running directions seen with 60-degree shifts and not for those with 45 or 90-degree shifts.
Oh, so this will confirm the proposed model of the equilateral triangle grid.
Yeah, and mean grid orientations vary between participants.
That will rule out activation as a result of visual cues.
Spatial memory was also shown to correlate with coherence, so the more participants were aligned with the grid cell's axes, the better subjects performed in the spatial memory task of replacing an object to its original location in the VR arena.
The model we created in the ROI approach was applied across the entire brain, and smaller populations of cells with the same six-fold rotational symmetry were found to a lesser extent in the other areas of the brain.
So, this may provide a cue for the neural basis of an autobiographical memory.
So, why do we care? fMRI is a much less invasive technique and can be used for the exact same purposes as single-cell recordings. Furthermore, we have now identified the presence of grid-like cells in humans. And finally, this is a really cool use of virtual reality in a psychology study.
However, there are some limitations. First of all, the equilateral triangle model is just a proposed model of how the brain represents space, and this may not be correct. Furthermore, the subjects controlled their position remotely using computer keys, and it may result in different pattern of activations than if they were actually moving. Lastly, only male participants were used to collect the data in this study.
The results of the study can also be applied to Alzheimer's research in the future. Because the entorhinal cortex is the bridge between the hippocampus and the rest of the neocortex, and in Alzheimer's there is shown to be degradation of the hippocampus, then this would help explain why Alzheimer's patients often explain feelings of disorientation. This means we could possibly use virtual reality to perform studies on Alzheimer's in the future.
The findings in this study may be used to inspire future research about more than Alzheimer's. Recall that evidence for grid-like cells with the same six-fold rotational symmetry was observed in brain regions beyond the entorhinal cortex. It could be valuable to explore the function of this network and determine whether the location of the grid-like cells is associated with different functionality.
The regions where these cells were found largely overlap with the network known to be associated with autobiographical memory. Therefore, this larger network may reveal how place and time context is encoded for long-term memories in the entire brain.
Now you know. Beyond using Google Maps and consulting your cell phone, you may have an internal GPS-like system that can help you remember where you have been before.